Thanks for the introduction, and hi everyone. Thanks for having me today. I'll share my perspective of working as a data scientist in the field of the big pharmaceutical industry. And for people that, I can skip that, for people that don't know what Novartis is, it's one of the top big pharma companies, and we work on developing innovative medicines across multiple diseases. So today I will talk about three main topics. So first will be more a kind of an introduction around drug development with the eyes of a data scientist, and also talk a little bit about what type of data we deal with on a day-to-day basis. And then I'll dive deeper into two use cases. These are really two practical projects or examples that they have in common: the monitoring of mobility across different diseases and how we can use it in our clinical research. And last but not least, I'll share maybe some challenges and key takeaways from my own experience as a data scientist in the field. So let's start with some introduction. I don't know how many of you are familiar with the process of drug development. It's quite long, it's complex, and it's highly regulated. And it usually starts by looking for a target, which is a gene or a protein in the human body that is known to be connected to a disease or multiple diseases. And then it's all about discovering a molecular structure or a drug candidate that will have the desired effect on that target and therefore maybe improve the course of a disease. And if everything seems to be okay, so we find some potential drug candidates, we run some simulations, work on cell lines, we do animal models. And then after that, if everything is safe and seems promising again, we move into human research. So we conduct different phases of clinical trials to assess the safety and the efficacy and effectiveness of this drug on real patients. Then, if all the results of these stages are positive, we collect all our research, all the data, submit it to health authorities like the American FDA. And after this approval, we can finally launch the drug on the market, so it will end up on the shelves of a pharmacy. Now, this process is very long, is very expensive, and very unsuccessful, so it sucks. It's long because it usually takes, for industry average, 10 years to start from a drug candidate all the way to have the drug in the market. It's expensive because nowadays, I think it reached around $2.6, $2.7 billion per drug, and it's unsuccessful because there's a huge failure rate. We start with thousands of drug candidates, maybe 100 makes it to the preclinical phase, and only one will reach the market. So it's quite of a tough business, but why do we like it? Why do we keep working on that as data scientists? Well, of course, beyond the fact that it's very rewarding that you're contributing to improving the health of patients. There are nowadays three types of innovations that I think are quite cool and quite interesting to look at. The first one is related to data. Recently, we've seen a lot of, especially accelerated by the pandemic, an explosion of the healthcare ecosystem, the type of data we can collect, the accessibility to this data, the volumes, and so on. The second one is models, so the advances of machine learning and AI. I'm not the one here that will say how much are these advancing and create new transformations. And especially in the pharma industries, we've seen that, for example, generative AI or LLMs, they're already really changing a little bit how we develop drugs. So for example, generating new molecular structures, identifying in a more automatic way the interactions between proteins and the drugs, understanding how we can better leverage single-cell data, very high-dimensional data. All of this is really shortening. I think this is the main point, shortening developing our drug. And I think there's already some early proof that from average 10 years, we can shorten to 7, which is still long, but much better. And then the third concept is precision medicine, or more broadly, personalized care, where instead of developing one pill that fits all, what our sales department would like to have, instead, the idea is that we would tailor drugs to specific patient groups or even specific individuals to deliver the best possible efficacious and safe care to patients. So for now, I want to take a little digression and talk a little bit more about data, because I think knowing what type of data you deal when you are in the pharmaceutical research really helps, and for data scientists to think about what strategies, what approaches, what methods, and anticipate challenges. So one fascinating thing is the multimodality. To really understand what a disease causes and what is caused by a disease and what happens in our body, you really need to look broadly and think about all different possibilities. From the molecular level, really looking at what we call omics. So it's omics data are profiling data. They look into what type of proteins you have in cells and tissues, what type of transcriptome, and on the genetic side, of course. And then you also look at imaging, and imaging are also different scales. So you have high-resolution imaging coming from histology, so pieces of tissues, all the way to microscopic images like MRI, CT scans, ultrasounds. Maybe some of you are familiar when you go to see a healthcare practitioner. There is another category which is more recent, I would say, and this covers the digital health data. These are data coming from your Fitbit, your Apple Watch, other type of sensors, ambient sensors, as well as more traditional medical devices like ECG. And the fourth category are electronic records, so doctor notes and traditional laboratory values which are conducted in normal clinical practice. And I think today there was also a workshop around how to mine electronic records data. In the examples that I will share later, just keep in mind this digital health data, because I will come back to that. There are also a couple of points to consider when we look into differences between data coming from the traditional tech world or tech applications and what happens in biotech or healthcare or pharma. So I mentioned about the different modalities, but also we are looking at different volumes of data. This doesn't mean that data sets in health are necessarily very small, but they are definitely different. So you might be working with this ultra-rare disease where there are literally like a dozen of patients worldwide, versus you're working with a disease that is more common, like we saw examples earlier today around diabetes. Also, feature space is very different, and in the healthcare space, due to also the different modalities, it can be several-fold larger. The other part is around annotations. So usually, and maybe I'm oversimplifying here, but events that you want to predict or you want to target in tech applications are relatively simple. You think about engagements, so number of clicks, downloads, screen time, etc. But if you're thinking about healthcare concepts, sometimes it's even hard to translate a clinical concept into an objective measure. And on the other hand, you also need the experience of one or multiple clinicians or biologists, so really domain experts to obtain this. And the last part is around access to data. So healthcare data is highly regulated. There are a lot of rules, regulations. They also depend on the geography where you are. And also for each data piece, we need to have informed consent from the patient. It has to be approved by an ethics committee. So every time you want to set up a study or a project as a data scientist, you need to double check all of these pieces are in place. And also, if you want to acquire new data, it can be quite expensive as well. Okay, I think that's enough about kind of introduction. So let's dive deeper into, as a data scientist, some use cases and concrete examples. So I mentioned at the beginning the drug development process. So one of the bottlenecks is when you move from bench to bed. So basically when you're moving from experiments on cell lines, animal models, and you move into patients. And this is the phase that we call translational medicine. And this is also the field where I mostly work on. And today I want to share basically what does it mean in practice as a data scientist if you're working in translational medicine, considering some of these new innovations, such as increasing amount of data, concept of precision medicine, and so on. So literally, the key scientific questions that we usually address can be categorized in 3 buckets. There's also, of course, more to that, and I will be very happy to talk about it offline after. But these 3 categories that I've listed here are around, the first one is on patient groups. So For example, can we identify different stages of the disease? Or can we identify patients that progress faster versus patients that progress slower? And what are the key measures that are driving this change into the disease stage? Or we want to identify specific, molecularly driven, what we call endotypes, so patient subgroups that have a very specific different manifestation at the molecular level. The other part is developing new measures. Of course, coming from a pharma company, the measures that we want to develop, the context of use is always to basically when we have a clinical topic that we want to measure and we don't have adequate means to do that, then we're interested in finding something new. And the use would be to show superiority of our product compared to the competitors or compared to the standard of care. So also questions regarding developing these new measures come quite frequently. And the third bucket is around proving drug effect. So which effects a new treatment is causing on patients, what changes, how they relate to safety signals, how they relate to the initial knowledge of the mode of action of the drug, but also how they relate to how the patient is feeling or what are the current common clinical measures of the disease progression. So maybe the third bucket covers a high percentage of the work I do and my team does, but I can't talk a lot about that because, of course, it's all IP protected. So today I will talk a little bit more about the first two parts around patient groups and new measures, and I will use a common example about how mobility that is measured commonly using sensors like accelerometers or gyroscopes, so what we call IMUs, inertial measurement units, can be used to advance our clinical trials. So let's start with the first topic. And the question that we're going to address is how can we meaningfully and accurately quantify patient mobility in real-life settings? So mobility, which is how well we move or how well we walk, is a very important quality of life factor. And it's unfortunately impaired in many diseases and in very different ways. Think about a Parkinson's disease patient, multiple sclerosis one, or somebody that broke the hip or had a stroke. So there are different types of motion impairment. And especially when we want to measure mobility in real-life settings, this is not quite easy. And an ideal solution basically would be, how would we do that? We would want something that is not expensive, that allow us to continuously measure the patients, so 24/7, really to have a comprehensive view. It should be something that does not add on to burden to patients as they are dealing already with their disease. And at the same time, something that from a methodology point of view, so an analytics point of view, should be robust to different type of motion impairment, but also should read out with a measure that is of importance for patients on the specific disease. So the current standard nowadays for this mobility, real-life mobility monitoring, is to use sensors that I mentioned before, like Fitbits, or your Apple Watch, other smartwatches, and their more clinically used versions like Actigraph or other brands like this. And of course, they fulfill the first 3 points. However, there's still quite some work to be done to be robust to different type of motion impairment and also to be with a context of use which is closer to what the patient needs. And this is exactly the aim of one of the IMI initiatives. This is Innovative Medicine Initiatives, are industry, academia collaborators, mostly sponsored by the EU. And the specific one is called Mobilize-T, where we are also part of, and my colleague Felix is driving one of the work packages there. So I will talk a little bit more about this work. So for this technical validation study, what we really wanted to do was to see if we can reliably extract gait in real-life settings or free living settings using wrist-worn IMUs, so wrist-worn sensors, which are the one most accepted by patients. So we set up this study where we had a body sensor network, consisting on insole sensors, a sensor strapped to the foot of the patient, on the lower back, which is currently also used in clinics, and then our target, which was the sensors worn at the wrist. And then we asked patients kind of to keep this sensor network on and conduct for 2 to 3 hours their standard activities. We collect all this data from a relatively small pool of patients and healthy controls across different diseases. And then we collected all their data, and we basically used the reference sensors, the insoles and the foot one, to obtain our gold standard. So really there's plenty of standard algorithms to detect the parts where patients are walking versus just normal movement. And then we took the wrist-worn sensors. These are 3D accelerations. We computed the magnitude of the vector, and for us, patients look something like that, kind of a noisy type of time series. And what we want to do is we want to automatically detect the segments where a patient is working and identify also what's the best approach to do that is as close as possible to the gold standard. So what was done in this work, which will be hopefully published very soon, so there's a reference at the bottom. A bunch of approaches were benchmarked. These are a combination of methods that are more time series based, like for example, based on wavelets, versus traditional machine learning ones, and also deep learning ones working on raw data. And we identified that actually the best performing method was this one published by Brand et al. Last year. It's a convolutional network-based method. And the performance metrics that we use is a combination between accuracy, sensitivity, and specificity. This is explained in the paper. But then when we looked more in details, in this specific case, we looked at the sensitivity, we saw that there were still a few outliers. Now, the method performed pretty well, still very, quite robust, but we need to be almost perfect. If you want to use it for clinical application, it has to be robust. So then we kind of looked, okay, what is driving these outliers here? And basically, we looked at, the best cases. Okay, this looks pretty good. We can segment gait very well. And this was also a subject that didn't need, for example, any walking aid. So had some mobility issues, but still could manage by their own. Then we looked at a case that is very close to the median. And surprisingly, we saw there was a patient where the method performs very well that was using crutches. So still, okay, we start gaining some confidence. But then when we looked at one of the cases with very low sensitivity, we saw very bad performance, if you can see there. And that patient used a bilateral walking aid, so something looking like that picture where you have a rolling device that really helps you kind of shuffle in the front. So that means that basically for us, as a lot of patients actually, they need to use these devices, we are not quite there yet into using gait really reliably for our clinical studies. So what is happening with part of the consortium is really try to then improve the CNN approach. So the convolutional network approach was still the best performing one. And also by collecting more and heterogeneous data and very well annotated. So just to be 100% sure that it's not a problem only on the labels from the reference systems. Now, why a company like Novartis is interested into that, and how are we using this type of research, this type of piece into our business? So we are definitely now, since a few years now, implementing and deploying these wrist-worn devices in our studies. We do it in two settings. We have a more complex setting for in-clinic motor function assessment, and then we have another solution, which is then we ask patients to continue wearing the wrist-worn device for the whole duration of the study, which is like 3 to 9 months, depending on the disease. And one of the interesting use cases, which we'll read out in maybe 1 or 2 years, will be on knee osteoarthritis. So we have several studies conducted on that. And then the idea is basically when we get this type of data, we extract the gait in the free living, and we then look at changes compared to the baseline visit before they started a new treatment, and see how these parameters of gait really change in time due to treatment versus the placebo group, or how well they are associated with other readouts which are on one side close to the mode of action of the drug. So for example, changes in specific proteins, for example, in the synovial fluid of the knee, versus also how the patient is feeling about their mobility. So it's kind of works very well in between the two evidence, from the subjective to the molecular space, and in a much more objective way than a clinician grading the changes in activity in a short time during an in-clinic visit. So let's talk now on the second example, which is on patient groups. So the question here that we want to address is, can we identify subgroups of patients at different stages of neurodegeneration? So here we're talking about Alzheimer's disease. And I want to start with a story of a patient. And she experienced some early symptoms, like a bit of memory loss, feeling herself lost sometimes and wandering, some balance problems, taking a lot of time to do some normal tasks. And of course, that worried her. So she was advised to go see a neurologist and underwent a battery of psychometric tests, which are normally used for diagnosis of Alzheimer's disease. And these tests cover a variety of aspects, such as memory assessment, orientation, attention, also language, motor skills, and so on. And then actually she scored pretty high. So doctors said, no, you don't have Alzheimer's, all good. However, that didn't stop her symptoms. So what is the problem here? On one side, the progression of Alzheimer's disease happens on a very long time frame. And it starts with a very slow cognitive degeneration in what we call the preclinical Alzheimer's disease. And then there is a phase where you basically starts to have cognitive decline until then it really rapidly evolves into Alzheimer's disease. And at the same time, the instruments that we have in the clinics for diagnosing Alzheimer's are done maybe once a year. They're quite subjective. It's a long set of tests. And they are really not sensitive to this early onset of cognitive decline. So then the main question is that could we use also this type of technologies like using sensors and digitalized tests to have a better assessment of the disease progression and categorize patients in the phase where they are currently are and see how fast or slow they progress. And this is the topic of yet another IMI initiatives called RADAR-AD that we are also part of. And the work that I'm going to share now about our contribution to this project was done by my colleague Gaetano. You can see the full name there, so if you have questions, you can also contact him directly on LinkedIn. And what was done in this consortia was to set up an observational study of 220 subjects, also not so large. There was unhealthy control and patients of different phases of Alzheimer's disease before the full onset of the disease. So we are talking about pre-Alzheimer's, prodromal, and mild Alzheimer's disease. And there were a battery of tests, of course, done. And the one that I'm going to talk today about was patients were basically, again, strapped with sensors on the lower back and the foot. And they were asked to do a simple test, which is first they walk up and down with a turn. You see the image on the lower part of the slide. Just walk for a minute, very simple, following the path. And then the second repetition is they need to count backwards while doing the same walking. So it's called dual tasking, which simulates actually a lot of things that we do on the day-to-day life, like you walk and tap on your phone, or you're carrying something, etc. So in this specific case, we asked patients to count backwards by 1, starting from 100. And we then looked at the raw acceleration data and the raw angular velocity of the sensors. We also had the aggregated overall gait parameters coming from the vendors that produce the sensors, and had available also the clinical test. So the main idea was that if we use the raw sensor data, can we improve this diagnosis that is done by multiple experts looking at this battery of tests and the clinical data and so on. So for that, we had a, by the way, spoiler alert, didn't work. But what we worked on was basically had an analysis pipeline where we did some, of course, pre-processing and then used both a supervised and an unsupervised tests to really see what was the role of gait with cognitive decline. So the pre-processing, very standard. Basically, we fragmented the time series into smaller chunks, did some normalization, etc., and also augmented the data because we had only 220 patients in different ways. The supervised approach, again, we benchmark a few approaches, and also by changing the type of input data that we were using. So, and then looked at in this particular case, we started with the area under the curve. And what we observed is that the best performing method was expert fully convoluted network. And by expert, because the network basically has an initial layer where it learns separately from the single task, so just walking, and the dual task, walking and counting backwards. And then the clinical parameters are added later on in the network. And this method performed the best compared to if we just use the normal clinical data that clinicians would use, or just by comparing with other more standard machine learning approaches. And yet, performance is really not so good, so it's only 0.75. And the task in this case was basically the classification if you are in the 4 categories: healthy, preclinical, prodromal, or mild AD. So then when we looked at the confusion metrics, we actually saw that a lot of these results were skewed by the accuracy on the healthy control country control class. And what we saw is that the network was really not good in distinguishing the last 2 phases so when you are from prodromal moving into mild Alzheimer's disease. So then, puzzled by that, we said, okay, let's remove that. Let's think about, we get the same data, and we just use an unsupervised approach. We only look at gait. Let's see if, in an unsupervised way, we see anything that is related to cognitive decline. So this is what we did. We basically used, in this case, a contrast learning approach, where we try to maximize the similarity for every single patient when they're doing a single task versus dual task. And then use the embeddings from this approach, and then cluster that. So what we found out were very complex results to analyze, or to interpret mostly, where we saw that actually the 3 obtained clusters could not very well distinguish the phases of Alzheimer's disease. So there was one part where maybe one group had the more severe, like prodromal and mild Alzheimer's disease, but it wasn't like a fine line. And then the second part, maybe reassuring, is that we did not see any differences, statistical significant differences when we think about them, like demographics, like sex, age, and so on. However, we saw that actually we could very well distinguish what we call gait behaviors. So there's a plot of one of the gait parameters, like cadence. And actually, these clusters, what they do very well is that they group different type of walking behavior. So maybe somebody walks a bit asymmetrically versus fast, and so forth. So now we are looking a little bit more in details on these type of clusters. But kind of as an outlook from this project, which involves so many partners and was very long, was that, yeah, maybe gait is not the best, or definitely taken in isolation is not the best for distinguishing the different phases of cognitive decline. However, it does help. So if we don't consider that, we are not as good as we should be. The study had a lot of limitations. Of course, the data were unbalanced, and also the task that we give patients maybe was too easy. We asked them to count backwards by 1. We could have done maybe a different setup. And also, we had no recordings on how they performed on the counting backwards. Maybe some patients made mistakes, or they repeated the same number twice, and so forth. So I think these type of measures really could help us. And this is exactly what we are now doing. So when thinking about this influence of motor and motor cognitive interference in our Alzheimer's disease trials. So we have worked, by we, I mentioned mostly Jelena and Jennifer, my colleagues that are our cognition experts. We have implemented a slightly different framework for dual task paradigms, so they're like more complex tests. And also we focus a lot on this motor cognitive interference that I just mentioned, rather than just looking in isolation at gait or cognition. And also beyond that, we also try to use voice as one of the new upcoming interesting markers. So feel free to contact them or ask me later like more about this type of work. Okay, so this basically brings me to the end of the examples. And for this last bit, I wanted to kind of share some takeaways, do's and don'ts, challenges and aspects that kind of we deal with every day when we work as applied data scientists in pharma. So when we think about data, the first thing that comes into mind is data biases. So it's so important for us to, every time we get a data set, to look into what type of potential biases there might be. And this can be maybe an ethnical group is not present in the biobank that we got from some Nordic countries. Or it can be we are totally looking only at younger patients and discarding certain age ranges. So it can be a variety of different biases. And it's very important that if you can't compensate for them, that you are super transparent about them, because if you're then using that solution in a global clinical trials, this might not work well. The second part is, and this is mostly when we want to have new measures and we want to acquire prospective data set, we need to be patient-centric. So patients are already dealing a lot with the burden of their diseases, so if we want to, in the case of gait, if we wanted to monitor their motions, we cannot really ask them in free living to wear, sensors of like 10 accelerometers strapped everywhere in the body. So we need to be very conscious about this part. And then the last piece, and Jocelyn earlier alluded to that, is the technology piece. For example, a version of a proteomic platform changes once a year. The firewall or like any sort of update on, software update on sensors every 6 months, maybe sometimes even less. A clinical trial lasts 2 to 5 years. So when we develop any solutions that is based on analyzing that type of data, we really need to be agnostic to this type of aspects, but also monitor it in the background. Is this update changing? Like, can I reproduce my results? What are the main changes and so on? So this adds a lot of work to a final model or a final solution. Then when we talk about analytics, the first thing is start from the main key scientific questions. So what is the need, the clinical need, the main questions, rather than, oh, I saw this like cool latest, Palm 2, I want to use it. Especially when we're talking to like applied data scientists, it's very important to start solving the problem that you have in hand, rather than using this like, a cool new method. And there are two aspects, especially when you are working into developing a machine learning solution, that really need to be, first in your mind. One is interpretability. The people that are going to use your solution are, patients, clinicians, biologists. They want to know what's happening. So when you co-create with them interpretability, or starting with very simple interpretable model, it's a very good start. And then you build on complexity and go to more sophisticated solutions where you could maybe give up some of the interpretability, but you've proven that the solution works. And then the last part is validation. And by that I mean, of course, benchmarking. You see that in these projects we always benchmark a lot of models and looking at what works best. The other part is around technical validation, but then clinical validations when you go into patients. So this is kind of like adds on many steps to make sure that your model works and your solution works. And the last part is around impact. In working in industry or a business like pharma, impact, so data science is not the core business. The core business of a pharma company is developing a drug. So as data scientists, we kind of contribute to that mission. So for us, when we are generating impact, it means that important decisions can be taken by using our results or our data. So to do that, it's very important that you work very closely with your collaborators that come from different domains. almost every project I work in, the core team has not only data scientists and engineers, but it has a biologist, a clinician, and then maybe a product person. So by working with them, like even in the co-creation and step-by-step, you really can make then a solution that then gets implemented, and the same stakeholders you have will take decisions based on your data. And the other part is that the healthcare and pharma industry especially, they are super highly regulated. So it's very important that we leverage a lot these pre-competitive collaborations with academia, with hospitals, to then start engaging with health authorities much earlier. Every time we want to maybe push a machine learning or AI solution applied to healthcare, if you do it not as a one player, but with a whole community and very solid research, then there's the path forward is kind of simpler. So you don't end up just having your amazing model, works very well, but just sits in a shelf and nobody uses it. I think in general, there are a lot of innovations coming from also different type of industries, and there's a lot of potential to really better understand like diseases and how we can better treat patients, really cure diseases, not only tackle some of the symptoms. So we really need more amazing data scientists to join this industry. Feel free to look up also at the Novartis website or connect with me on LinkedIn for like opportunities, etc. With that, thank you.